We'll resume from the last point. Now we have seen what is meant by Km. Km is nothing but the substrate concentration required to produce a half of maximum velocity, and it is a specific characteristic of a particular enzyme. It is a specific characteristic. मतलब हर एक के लिए Km वैल्यू क्या है, हर एक एंजाइम का स्पेसिफिक कैरेक्टरिस्टिक फीचर होता है. Now a low Km value, अगर कोई एंजाइम कैटालाइज रिएक्शन में if a low Km value in the enzyme catalyzed reaction that indicate there is a strong interaction between enzyme and substrate. A low Km value indicate the strong affinity between enzyme and substrate, whereas a high Km value or large Km value will reflect you that there is a weak affinity between your substrate and enzyme. Okay, so you have to remember the significance of low Km value and high Km value. इसका significance आपको याद रखना है. Low Km value means there is a strong affinity between enzyme and substrate. High Km value means there is a weak affinity between substrate and complex. Now most of the enzymes they will have Km value in a range of this 10 raised to minus 5 to 10 raised to minus 2, and it is a not Km is not dependent on concentration of enzyme. So Km का specific characteristics क्या होता है? So that is also important. You have to remember. Km is nothing but the Michaelis Menten constant or Briggs Haldane constant. It is also known as the Briggs Haldane constant. We have seen in the previous slide. इसमें देखो आपने ये देखा है Briggs and Haldane's constant, that is Km, or it is also known as a Michaelis Menten constant. Okay, remember this. And the value of Km is a specific characteristic for particular enzyme. Low Km value means there is a strong interaction between substrate and enzyme. High Km value means there is a less interaction or there is a weak interaction between enzyme and substrate. And most of the enzymes will have Km value in a range of 10 raised to minus 5 to 10 raised to 2. And Km value is not dependent on the concentration of enzyme. There is another plot known as a Line-Weaver-Burke plot or Line-Weaver-Burke plot. Line-Weaver-Burke double reciprocal plot. If you plot a reciprocal of your Michaelis-Menten equation, तो Michaelis-Menten equation है उसका reciprocal आप plot करेंगे तो you will get a Line-Weaver-Burke double reciprocal plot. So इसके ऊपर GPAT 2018 में क्वेश्चन आ चुका है. By taking the reciprocal of Michaelis-Menten equation, that is a reciprocal of velocity. पहले क्या था? Velocity is equal to Vmax S divided by Km plus S. ऐसा equation आपका Michaelis-Menten का. अभी क्या करना है? उसको 1 divided by करना है. So reciprocal of Vmax. So 1 upon V versus 1 upon S. If you take a reciprocal of velocity versus reciprocal of substrate concentration. So again, if you plot the graph, you will get a straight line. That is known as a Line-Weaver-Burke reciprocal plot. So there are some drawbacks of your Michaelis-Menten equation. So that's why. So this reciprocal plot or double reciprocal plot will give you idea regarding the enzyme inhibition and all that. So you can see over here. So if you take a reciprocal of your Michaelis-Menten equation, you will get a such kind of equation. 1 upon Vmax is equal to Km divided by Vmax into 1 upon S into 1 upon Vmax. Likewise, this is known as a Line-Weaver-Burke double reciprocal plot. So ऐसा इक्वेशन दिया था आपको GPAT 2018 में, ऐसा इक्वेशन दिया था और नीचे ऑप्शन में था ऊपर क्वेश्चन था आइडेंटिफाई लीन-वियर डबल रेसिप्रोकल. नहीं, ऐसा क्वेश्चन नहीं था. तो ऐसा क्वेश्चन था कि इफ यू प्लॉट a reciprocal of maximum velocity to the reciprocal of substrate concentration in enzyme catalyzed reaction, so following graph will be obtained. क्वेश्चन ऐसा था. एंड उसके नीचे ऑप्शन थे आपके Michaelis-Menten प्लॉट, लीन-वियर-बर्क डबल रेसिप्रोकल प्लॉट और भी एक दो ऑप्शन थे. सो आंसर वाज अ Line-Weaver-Burke double reciprocal plot. So if you plot a reciprocal of substrate concentration versus reciprocal of maximum velocity, you will get a Line-Weaver-Burke plot. You will get a straight line. You can see over here. So this is a Line-Weaver-Burke plot, straight line. So this is x-axis substrate concentration, y-axis है maximum velocity. Y-axis है maximum velocity. Now this is known as a maximum velocity. Now line behavior work double reciprocal plot will give you more idea. So its x intercept is a 1 upon km. यहाँ पे जो x axis पे इंटरसेप्ट मिलेगा आपको उसको बोलेंगे 1 upon km. And the slope of this, इसका जो slope है, slope of this graph is nothing but the km divided by vmax. और इसका स्लोप निकलेंगे आप ऐसे, स्लोप जो है इट इज अ km डिवाइड बाय vmax एंड y इंटरसेप्ट क्या है 1 अपॉन vmax, y अपॉन vmax. देखो y इंटरसेप्ट कहाँ पे है, x इंटरसेप्ट है 1 अपॉन km, y इंटरसेप्ट है 1 अपॉन vmax एंड स्लोप क्या रहेगा vmax डिवाइड बाय km डिवाइड बाय vmax. That is known as a line behavior work double reciprocal. Double reciprocal the double reciprocal plot is useful in understanding the effect of various enzyme inhibitors. But your Michaelis-Menten equation was not giving sufficient information regarding the activators and inhibitors. Okay, so that is the main difference. सिर्फ इक्वेशन याद रखो, लाइन बियर वर्क डबल रेसिप्रोकल प्लॉट में क्या है कि 1 अपॉन वीमैक्स है, यहाँ पे रेसिप्रोकल है, मतलब रेसिप्रोकल मतलब क्या है 1 अपॉन वीमैक्स. और माइकलिस मेंटेन इक्वेशन जो है दैट इज vmax इज इक्वल टू 1 बाय जो है ना vmax इज इक्वल टू v इज इक्वल टू vmax इनटू s डिवाइड बाय km प्लस s. ये आपका माइकलिस मेंटेन है और उसका ही रेसिप्रोकल लेना है, मतलब 1 अपॉन v इज इक्वल टू 1 अपॉन vmax, ऐसा ये प्लॉट इक्वेशन आएगा. नाउ these two equations, line behavior work double reciprocal plot and Michaelis Menten equation, they are known as the enzyme kinetics. Now next one is what is the effect of temperature on the enzyme catalyzed reaction. So as you increase the temperature of a reaction, so reaction velocity will be increased. So increase in the reaction velocity of reaction is increased with increasing the temperature up to maximum temperature and then declines. इनिशियली जैसे टेम्परेचर इंक्रीज करेंगे तो वेलोसिटी इंक्रीज हो जाएगा अप टू ए सर्टेन टेम्परेचर, और उसके बाद में क्या होगा वेलोसिटी कम हो जाएगा. ए बेल शेप्ड कर्व इज ऑब्टेन्ड, ए बेल शेप्ड कर्व इज ऑब्टेन्ड, यू कैन सी ओवर हियर. सो इफ यू टेक टेम्परेचर x axis and velocity on y axis, so if you increase the temperature, so increase in temperature initially will increase the rate of reaction or velocity of reaction at a certain temperature and then decline. Now this temperature at which enzyme will show highest activity, that is known as a optimum temperature. So temperature coefficient or q10, temperature coefficient or q10 is nothing but the increase in the velocity of enzyme when temperature is increased by 10 degrees Celsius. So q10 temperature coefficient is defined as the increase in the velocity of enzyme when there is increase in the temperature by 10 degrees Celsius. That is known as Q10. And for most of the enzyme, Q10 is between 2 to 40 degrees Celsius. बहुत सारे एंजाइम इसके बीच में ही टेम्परेचर में काम करते हैं. But there are certain exceptions. For example, tag DNA polymerase. So that will have an optimum temperature of 70 degrees Celsius. Then muscle phosphatase, that is also having temperature more than 40 degrees Celsius. So optimum temperature, most of the enzymes they will have optimum temperature in the range between 2 to 40 degrees Celsius. But there are certain exceptions, as I have told you, tag DNA polymers that have optimum temperature more than 70 degrees Celsius, and muscle phosphokinase that also muscle phosphatase that will also have optimum temperature more than your 40 degrees Celsius. In the similar manner, if you take a pH, so enzyme catalyzed reaction, rate of reaction or velocity of reaction versus pH. So by increasing the pH or hydrogen ion concentration, there is increase in the velocity or rate of reaction. So increasing the pH, the velocity or rate of reaction will be increased at a certain certain range of pH. After that it will decline. So initially जैसे आप pH increase करोगे, enzyme का velocity increase हो जाएगा at a certain level और बाद में decrease हो जाएगा. Again you are getting a bell shaped curve. The pH at which enzyme will show its highest velocity, that is known as the optimum pH. जैसे optimum temperature होता है वैसे optimum pH होता है. Effect of pH on enzyme catalyzed reaction. A bell shaped curve is obtained. Each enzyme will have optimum pH, ठीक है, at which the velocity is maximum. The pH at which enzyme will show its maximum velocity in enzyme catalyzed reaction, that is known as the optimum pH. Below the optimum pH, the velocity will be less or will be decreased, and beyond the optimum pH again velocity will be decreased. तो velocity जो है reaction का वो optimum pH के वहाँ पे ही optimum pH पे ही ज़्यादा रहेगा, उसके नीचे कम रहेगा, उसके ऊपर भी कम रहेगा. Now effect of product concentration, जैसे product concentration जैसे बढ़ रहा है, जैसे 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 product बढ़ेगा, so if you plot a reaction velocity versus product concentration, as there is increase in the product concentration, so velocity will be decreased. Okay, so increase in the product concentration will decrease the reaction velocity. Okay, these are the effect of different factors on enzyme catalyzed reaction. What is the effect of enzyme concentration? What is the effect of substrate concentration? What is the effect of temperature? What is the effect of pH? What is the effect of product concentration? So you have to remember this. Now, some enzyme require certain inorganic metallic cations like magnesium, manganese, zinc, calcium for their optimum activity. For their optimum activity. So they are known as metalloactivators. So activators, enzyme activator. So some enzyme require these cations. आपने देखा होगा कि कोई रिएक्शन में Mg प्लस लग रहा है वगैरह. So these are known as metallic activators, and but these metallic activators they are not bound to your enzyme. जैसे enzyme को ये covalently bind नहीं करेंगे, so they are known as a metallic activator. These ions they bounce enzyme covalently and they are a part of enzyme only. Then they are known as a metalloenzymes. So metalloenzymes hold the metal tightly, which are not readily exchanged. Okay. Now metallic activator और metalloenzyme ज़्यादा डिफरेंस है नहीं है, कौन सा है, थोड़ा डिफरेंस है. मेटेलिक एक्टिवेटर्स क्या करेंगे, वो एंजाइम को एक्टिवेट करते हैं, बट दे आर नॉट अ पार्ट ऑफ एंजाइम, जैसे मैग्नीशियम एमएन प्लस जिंक वगैरह. बट मेटेलो एंजाइम इज दे आर अ पार्ट ऑफ एंजाइम, they will form a covalent bond with the enzyme. Now enzyme inhibition. So what is meant by enzyme inhibition? So decrease in the catalytic activity of enzyme. Decrease in the catalytic activity of enzyme, that is nothing but the enzyme inhibition. ये enzyme का काम नहीं करने देना, उसको बोलेंगे enzyme inhibition. एंजाइम को इनहिबिट करना मतलब एंजाइम को अपना काम करने से रोकना. So enzyme inhibition means decrease in the catalytic activity of enzyme, that is known as the enzyme inhibition, and they are of three types. So they are of three types: reversible inhibition, irreversible inhibition. First one is the reversible inhibition, second one is the irreversible inhibition, and third one is the allosteric inhibition. So we are going to see reversible inhibition and irreversible inhibition. So reversible inhibition means what? The inhibitor binds non-covalently with the enzyme and inhibit the enzyme. Now this as there is a non-covalent binding of inhibitor with the enzyme, it can be reversed. ठीक है, जैसे इनहिबिटर जो है वो नॉन कोवैलेंटली बाइंड करेगा एंजाइम को और एंजाइम का काम उसको काम करने से रोकेगा, लेकिन जैसे जैसे वो इनहिबिटर डिटैच होता है एंजाइम से, बाद में एंजाइम एक्टिवेट हो जाएगा. ओके, रिवर्सिबल इनहिबिशन मींस इनहिबिशन the inhibitor binds to enzyme non-covalently with the enzyme and inhibit the enzyme catalyzed reaction, and it can be reversed. Now these reversible enzyme inhibition are also again of two types. ठीक है, फर्स्ट टाइप है आपका कॉम्पिटिटिव इनहिबिशन. कॉम्पिटिटिव इनहिबिशन मीन्स कॉम्पिटिशन है, यहाँ पे कॉम्पिटिशन किसके बीच में होता है, दो के बीच में, लेकिन दो के बीच में भी होता है, लो ऐसा नहीं कि एक छोटा बच्चा है और एक बड़ा पहलवान है दोनों कुश्ती लड़ाओ, नहीं, कंपटीशन किस में होता है, साथ में के साथ, जैसे दोनों पहलवान का वेट अगर सेम है, एज उसी रेंज में है, तो मीन्स देयर शुड बी सिमिलैरिटी. नाउ योर कॉम्पिटिटिव इनहिबिटर विल हैव स्ट्रक्चरल सिमिलैरिटी टूवर्ड्स लाइक अ सबस्ट्रेट. जो आपका सबस्ट्रेट है उसके जैसे ही उसका स्ट्रक्चर रहेगा, उसका सिमिलैरिटी रहेगा. कोई कंपटीशन किस में होता है, दोनों साथ में अगर होगा तो उसका कंपटीशन. ऐसा नहीं कि आप फर्स्ट ईयर में हो, आप फर्स्ट ईयर बी फार्म में हो और एम फार्म सेकंड ईयर के बच्चे के साथ आपका थोड़ा कंपटीशन रखेंगे किसमें, फर्स्ट ईयर के बच्चे में रखेंगे. तो ऐसा कंपटीशन हैपेंस इन द कैंडिडेट्स हैविंग सेम फीचर. तो वैसे ही कंपटीशन किस में होगा, इफ योर सबस्ट्रेट and inhibitor is having similar structure, then only there will be competitive inhibition. Okay, so your inhibitor they will have close resemblance to substrate and can be regarded as a substrate analog. And in non-competitive inhibition, in competitive inhibition, remember this line. In competitive inhibition, Km value increases. In competitive inhibition, Km value increases and Vmax remains unchanged. यहाँ पे Km वैल्यू क्या होगा, इंक्रीज होगा. Km वैल्यू मतलब क्या, Km वैल्यू मतलब सबसेट कॉन्सेंट्रेशन required to produce half of maximum velocity. तो सबसेट कॉन्सेंट्रेशन यहाँ पे Km वैल्यू यहाँ पे बढ़ जाएगा और जो वेलोसिटी है दैट विल रिमेन अनचेंज. फॉर एग्जांपल क्या है कॉम्पिटिटिव इनहिबिशन का एंजाइम. फॉर एग्जांपल एंजाइम सक्सिनेट dehydrogenase is a classical example of this type of inhibition. So enzyme succinate dehydrogenase is a classical example of competitive inhibition. So for example succinate dehydrogenase क्या करेगा, इसका सबस्ट्रेट क्या होगा, सक्सिनिक एसिड. सक्सिनेट डीहाइड्रोजनेज का सबस्ट्रेट क्या होगा, सक्सिनिक एसिड. दिस टाइप ऑफ ओके. नाउ सक्सिनिक एसिड अगर होगा तो ये रिएक्शन दिखाएगा, लेकिन अगर मेलानिक एसिड है, मेलानिक एसिड और ग्लूटारिक एसिड एंड ऑक्सैलिक एसिड. सो मेलानिक एसिड, ग्लूटारिक एसिड एंड ऑक्सैलिक एसिड, these three are known as the competitive inhibitors of succinate dehydrogenase enzyme. So instead of succinic acid, this malonic acid and glutaric acid or oxalic acid, they will bind to your enzyme and they will inhibit it. Because these three, malonic acid, glutaric acid and oxalic acid, they have structural similarity with the your succinic acid. Okay? That's why there is a competitive inhibition. Another example, for example, methanol. Another example is a methanol. Methanol can be used in treatment of alcohol poisoning because methanol and ethanol is having similar structure. Now methanol can be used in case of alcohol poisoning because methanol and ethanol is having similar structure. So आपको मालूम है alcohol poisoning में कौन सा drug use करते हैं, that is nothing but the disulfiram. Okay, disulfiram, that is a alcohol dehydrogenase inhibitor. Okay, so ethanol or methanol can be used in case of ethanol poisoning. Ethanol can compete with the methanol. अगर आपको methanol का poisoning हो गया तो methanol को metabolize करने वाला enzyme कौन सा है? The enzyme is nothing but the alcohol dehydrogenase. Okay, so methanol and ethanol, these two have similar structure. Okay, that's why ethanol and can compete with the methanol for alcohol dehydrogenase enzyme. And methanol, instead of getting methanol metabolized, your ethanol get metabolized. That's why. So that this is known as a competitive inhibition. Competitive inhibition मतलब क्या, दोनों में structural similarity रहेगी inhibitor और substrate में. So your substrate as well as your inhibitor will have structural similarity and they will compete each other for a enzyme. That is known as a competitive inhibition. Okay. Now next one is a non-competitive inhibition. Next one is a non-competitive inhibition. Next one is a non-competitive inhibition. यहाँ पे देखो, यहाँ पे कोई competition नहीं है, किसी का competition नहीं हुआ. So in case of non-competitive inhibition, the inhibitor and substrate will not have any structural similarity. Okay, will not have any structural similarity. Now this inhibitor binds to enzyme to the other side. ठीक है, other side को bind करेगा. जैसे competitive inhibitors में क्या रहता है? So in case of competitive inhibition, so your substrate and inhibitor, they have competition at the active site of enzyme. एक्टिव साइट जो है एंजाइम का वहाँ पे ही कंपटीशन हो रहा है. इन केस ऑफ नॉन कॉम्पिटिटिव एंजाइम इनहिबिशन, ए इनहिबिटर विल नॉट बाइंड टू एक्टिव साइट, it will bind to another site. Inhibitor bind to another site than active site, interferes with the enzyme activity. This binding impairs the enzyme function and inhibitor does not have any structural resemblance to substrate but has a strong affinity for the enzyme. It binds to another side of enzyme. In this type of inhibition, Km value remains unchanged and Vmax is lowered. ये याद रखो. जैसे competitive inhibition में क्या होता है, Km value increases but Vmax remains unchanged. In case of non-competitive inhibition क्या होता है, Km value is unchanged and Vmax is lowered. ये दो याद रखो लाइंस, बहुत ही इंपॉर्टेंट है, आपको आ सकता है कुछ. Examples of this type of inhibition, for example, heavy metals. Heavy metals like your lead, mercury, argon, etc. They can bind non-competitively at the enzyme. They binds the enzyme non-competitively. Not they do not bind to active site. They binds to another site. They used to bind to these groups: cysteine group, sulfidyl group of the amino acid residue of protein, and they will inhibit the enzyme. That is known as a non-competitive inhibition. Now next one is irreversible inhibition. So in case of competitive as well as non-competitive, whatever binding of inhibitor with the enzyme, that was a non-covalent bonding. That was a non-covalent bonding. Now in this irreversible type of inhibition, there is a covalent binding. In case of irreversible inhibition, there is a covalent type of bonding. In case of irreversible inhibition, there is a covalent type of bonding. So there is a covalent bonding between your inhibitor and enzyme. So irreversible inhibitor मतलब क्या? Irreversible मतलब covalent bonding हुआ हुआ. So inhibitor binds covalently with the enzyme and inactivate the enzyme, which is irreversible. So inhibitors of such type are toxic substances, and these substances poison the enzyme. For example, pyruvate acetate is an irreversible inhibitor of enzyme like glycerylate 3-phosphate dehydrogenase, papain. Okay, they combine with the sulfidyl group of enzyme and inactivate them. Irreversible type of enzyme. So diisopropyl chlorophosphate (DEFP). It is again a irreversible inhibitor to a cholinesterase enzyme. Diisopropyl chlorophosphate is known as a nerve gas. ये अगर गैस अंदर गया तो कोलिनेस्टरेज एंजाइम जो है वो इनहिबिट हो जाएगा और वहाँ पे एसिटिल कोलिन का लेवल आपकी बॉडी में बढ़ जाएगा और वहाँ पे आप पेडिकार्डिया, ठीक है, चोकिंग वगैरह से आप फिर बाद में मर जाओगे. So diisopropyl chlorophosphate was the nerve gas developed by German during World War II. जर्मनी ने ये नर्व गैस डेवलप किया था ड्यूरिंग वर्ल्ड वॉर सेकंड. हिटलर ने इसको यूज किया था. And this nerve gas inhibits the serine proteases enzyme and acetylcholine enzyme irreversibly, and then आदमी मर जाता है. So it is an irreversible inhibition. There is a covalent bonding between inhibitor and enzyme. Okay. For example, another example, cyanide inhibit cytochrome oxidase. Okay, irreversibly. Cyanide is an inhibitor and cytochrome oxidase is an enzyme. So there is an irreversible inhibition. Again, disulfiram. It is a drug used in the treatment of alcohol intoxication. It is an irreversible inhibitor of aldehyde dehydrogenase used in the treatment of alcohol addiction. Our next one is suicide inhibition. Suicide inhibition. Now, इसमें क्या होता है कि it is a specialized type of inhibition where it is a specialized type of irreversible inhibition. In this case, original inhibitor is converted to more active or more potent inhibitor by the same enzyme. ठीक है, जैसे inhibitor है combine होगा enzyme के साथ में और ज़्यादा potent inhibitor बनेगा और ज़्यादा. For example, so it is a specialized type of inhibition of irreversible inhibition in which क्या होता है इसमें original inhibitor is converted into more potent inhibitor by the same enzyme. Okay, thought to be inhibited. For example, allopurinol. Allopurinol is a drug used in the treatment of gout. So it is a inhibitor of xanthine oxidase. ये एंजाइम को इनहिबिट करता है. सो एलोपुरिनॉल इसको इनहिबिट करता है. तो एलोपुरिनॉल जब इसके साथ में कंबाइन होता है, सो एलोपुरिनॉल इज कन्वर्टेड इनटू वन मेटाबोलाइट नोन एज एलोक्सेंथिन by this xanthine inhibitor. So allopurinol का conversion करेगा ये alloxanthine में, but this alloxanthine is a more potent inhibitor than allopurinol. In the similar manner, you can see thiofluorouracil. So it gets converted into fluorodeoxyuridylate. Okay, thiofluorouracil, which inhibit the enzyme thymidate synthase, and this thiofluorouracil is used in the cancer. It is anti-cancer agent. Now another type of inhibition is a allosteric inhibitor. Allosteric inhibition. Okay, this type of inhibition is or it is actually, you cannot call it as inhibition. Allosteric modulators or allosteric regulators. So there is another site on the enzyme. So एंजाइम में बहुत सारे साइट्स होते हैं. जैसे एक्टिव साइट मतलब क्या है? The site at which your substrate will combine with the enzyme and convert into product. Okay. At the active site means there is a specific sequence of amino acid. Okay. Or active site is a specific site of enzyme, and that's why enzyme has a specificity. But there is another site known as allosteric site. So at this site, allosteric inhibitors combines and regulate the enzymes in the body. आपके बॉडी में एंजाइम का रेगुलेशन करने का कौन काम करता है? एलोस्टेरिक मॉड्यूलेटर्स. एलोस्टेरिक मॉड्यूलेटर, they modify the enzyme action by binding with the allosteric site and regulate the enzyme activity. If you want to read details regarding this, you can read from Biochemistry by Sachin Jadhav. Now enzyme specificity. So enzymes are very specific in their action. Okay. So they have stereo specificity or optical specificity. They have reaction specificity and substrate specificity. Look, D-glucose will only metabolize the D-glucose, not L-glucose. D-amino acid oxidase will only cause oxidation of D-amino acids, not L-amino acid. So they have stereo specificity. Reactions oxidoreductase will only catalyze the reaction where there is oxidation and reduction. They will not catalyze the hydroxylation reaction. जैसे hydroxylase एंजाइम है तो वहाँ पे हाइड्रोक्सिलेशन होगा, वहाँ पे ऑक्सीडेशन रिडक्शन नहीं होगा. सो स्पेसिफिक दैट इज नोन एज द रिएक्शन स्पेसिफिसिटी. सबसेट स्पेसिफिसिटी मतलब क्या है? पर्टिकुलर सबसेट के ही रिएक्शन करेगा. जैसे जैसे अल्कोहल डीहाइड्रोजनेज, दैट मीन्स वो अल्कोहल को ही डीहाइड्रोजनेशन करेगा, वो एल्डिहाइड को नहीं करेगा, फिर एसिड को नहीं करेगा. So that is a substrate specificity. So enzymes will have stereo specificity or optical specificity. They are reaction specific. They are a substrate specific, etc. Okay. And remember, enzyme specificity is mostly dependent on the apoenzyme part of enzyme. यहाँ पे देखो, यहाँ पे लाइन लिखा है, enzyme specificity is mostly dependent on the apoenzyme, not on a coenzyme or cofactor. ये लाइन मैंने उस टाइम पे भी बोला when we was discussing about the, ये लाइन इंपॉर्टेंट है, इसके ऊपर क्वेश्चन आ चुका है. एंजाइम specificity is mostly dependent on apoenzyme, not on a coenzyme. इसके ऊपर भी क्वेश्चन आ चुका है. Now there are theories related to enzyme action. How enzyme will act on a substrate in an enzyme catalyzed reaction. उसके theories है. First one is known as a lock and key model. It is also known as a Fisher theory. And second one is known as a Coastlands model or induced heat theory. Coastlands model or induced heat theory. Enzyme activity is measured in case of one unit known as a catal. Enzyme activity measure करने का एक unit होता है known as a catal. KATAL. Okay, one molecule. Okay, and that is nothing but the enzyme activity is measured in the catal unit, and one cat is equal to one cat is nothing but the activity of enzyme that convert one molecule of substrate per second. Catal unit होता है enzyme activity का, याद रखो. One cat is nothing but the concentration of enzyme required to convert one mole of substrate per second into the product, and it is also international unit. One catal is equal to 6 into 10 raised to 6. ये याद रखो, 6 into 10 raised to 6. इसके ऊपर भी क्वेश्चन आ चुका है. One catal is equal to 6 into 10 raised to 1 cat is equal to 6 into 10 raised to 6. Okay. Then enzymes have different application in the industry, therapeutics, diagnostic application. Okay, we'll see one by one. This chapter is very lengthy chapter. Enzymes and their applications. Okay, so you can see enzymes, they are having numerous applications. Okay, they are used in the diagnosis, they are also used in the therapeutics, they are also used in the laboratory diagnosis. Okay, diagnosis, therapeutics, and industrial application. Okay, we'll see one by one. For example, you can see our clinical or therapeutic application of enzymes. So this streptokinase and urokinase, these are the two enzymes. They are used therapeutically. ये treatment के लिए use करते हैं streptokinase urokinase. So they are a plasminogen activator. यहाँ पे देखो PA लिखता हूँ मैं, PA. PA मतलब plasminogen activator. Okay. they are used to activate the plasminogen and break down the clots. They are known as a thrombolytics. They are known as a thrombolytics. They are used to dissolve the blood clots in the myocardial infarction, angina, and coronary clots. Then asparginase is the enzyme very important for the treatment of cancer. Then papin. Papin is an anti-inflammatory. Papin means it is a proteolytic enzyme, will break down the protein or clots and all that. जैसे आपका यहाँ पे अगर सूजन हो गया या फिर इन्फ्लेमेशन हो गया तो उस टाइम पे यूज करते हैं पैपिन. इसका टैबलेट भी आता है. अल्फा अमाइरिन एंड एंटीटिप्सिन. अल्फा एंटीटिप्सिन. So again used in the treatment of emphysema. Pancreatic enzymes, trypsin and lipase, they are used for digestive purpose. So these are the therapeutic application of enzyme. Few therapeutic application of enzyme. Diagnostic application of enzyme. Okay, diagnostic application. Diagnosis करने के लिए क्या application है? Laboratory. Okay, diagnostic application means laboratory estimation. So glucose oxidase and peroxidase, it is used in the estimation of glucose level in the body. So glucose oxidase and peroxidase enzyme, they are utilized for estimation of glucose. आपके बॉडी का ग्लूकोज चेक करने के लिए ये एंजाइम लगते हैं. Then urease is required for the estimation of urea. Cholesterol oxidase is used for estimation of cholesterol. Then uricase is required for estimation of uric acid. Lipase for triglyceride. Luciferase for uh, contamination, बैक्टीरियल कंटेमिनेशन इन द फूड. अगर फूड में बैक्टीरियल कंटेमिनेशन है तो उसको डिटरमाइन कर सकते हो आप बाय यूजिंग लूसिफेरेज. अल्कलाइन फॉस्फेटेज एंजाइम इज यूटिलाइज्ड फॉर एनालिटिकल टूल इन द ELISA टेस्ट. सो दीज आर द थेरेप्यूटिक एंड लेबोरेटरी एस्टिमेशन एप्लीकेशंस ऑफ एंजाइम. थेरेप्यूटिकली कौन से एप्लीकेबल है और डायग्नोस्टिकली कौन से एप्लीकेबल है. नाउ अनदर सिग्निफिकेंस ऑफ सम एंजाइम्स. ठीक है, कुछ कुछ एंजाइम्स ऐसे होते हैं कि कोई डिसीज में उनका लेवल बढ़ जाता है. सिग्निफिकेंस ऑफ सीरम कॉन्सेंट्रेशन ऑफ वेरियस डायग्नोस्टिक एंजाइम. सो दीज आर द एंजाइम्स, they are elevated, they are increased in case of myocardial infections or myocardial infection. अगर मायोकार्डियल हार्ट अटैक में ये एंजाइम का लेवल बढ़ जाता है, ठीक है, बॉडी में ब्लड में ये एंजाइम का लेवल बढ़ जाएगा ये हार्ट अटैक में. So we'll see that. First one is a myoglobin. Okay. First one is a myoglobin. So it is a early marker, but it is not a cardiac specific. Early marker, but not cardiac specific. Then cardiac troponin 1. ये enzyme है. It is a early marker and it is as well as cardiac specific. And this was question in GPAT 2020. Match the following type का क्वेश्चन था. नहीं सॉरी, डायरेक्ट क्वेश्चन था. वन ऑफ द फॉलोइंग हार्मोन इज अ अर्ली मार्कर फॉर डायग्नोसिस ऑफ मायोकार्डियल इन्फेक्शन. और आपके ऑप्शन में थे जैसे कार्डियक ट्रोपोनिन 1, देन लैक्टेट डिहाइड्रोजनेज, क्रेटिनिन फॉस्फोकाइनेज एंड और एक ऑप्शन था. ये तीन ऑप्शन थे, कार्डियक ट्रोपोनिन, लैक्टेट डिहाइड्रोजनेज, क्रेटिनिन फॉस्फोकाइनेज और एक ऑप्शन था. So remember, दोनों भी lactate dehydrogenase is also a marker for myocardial infection, but it is a late marker. lactate dehydrogenase जो है वो late marker है. अर्ली मार्कर कौन है? कार्डियक ट्रोपोनिन, GPAT 2022. तो कार्डियक ट्रोपोनिन सेकंड is also a early marker, but cardiac troponin second is also elevated in case of another disease. बाकी कोई डिसीज होगा तो उसमें भी ये बढ़ जाएगा, लेकिन कार्डियक ट्रोपोनिन फर्स्ट जो है दैट इज अ कार्डियक स्पेसिफिक and it is the early marker for myocardial infection. ठीक है, likewise that lactate dehydrogenase is a late marker but cardiac specific. ये cardiac specific भी है पर late marker है. creatinine phosphokinase is also cardiac specific and early marker. So cardiac troponin याद रखो, creatinine phosphokinase रखो. तो बहुत सारे बच्चों ने इनको इस क्वेश्चन के लिए ऑब्जेक्शन भी रेज किया था क्योंकि उसमें ये भी ऑप्शन था creatinine phosphokinase. क्रेटिनिन फॉस्फोकाइनेज इज आल्सो अर्ली मार्कर, तो कार्डियक ट्रोपोनिन भी अर्ली मार्कर है. ये दोनों याद रखो आप, इसके दोनों के ऊपर क्वेश्चन जीपैट 2022 में आएगा. नेक्स्ट विल सी एलिवेटेड लेवल ऑफ एंजाइम्स इन द हार्ट हेपेटिक डिसीज. लीवर डिसीज में कौन से एंजाइम बढ़ जाते हैं? तो फर्स्ट वन इज एलेनिन ट्रांसफरेज, ALT. सो इट इज इंक्रीज इन केस ऑफ वायरल हेपेटाइटिस, वायरल इन्फेक्शन अगर होगा लीवर का तो उसमें ये अलानिन अलानिन ट्रांसअमाइनेज, अलानिन ट्रांसअमाइनेज ALT, ये इसका लेवल बढ़ जाएगा बॉडी में. देन aspartate transaminase AST, its level is increased in case of C liver disease and obstructive jaundice. Then gamma glutamyl transpeptidase, gamma glutamyl transpeptidase, the level of this enzyme is increased in case of alcoholic liver disease. Iodase nucleotidase, so its level is also increased in case of hepatic cholestasis. यहाँ पे बाइल में अगर ऑब्स्ट्रक्शन हो गया तो ये लेवल एंजाइम का बढ़ जाएगा. सो दीज आर द एंजाइम यूज एंजाइम दैट दैट गिव सम आइडिया रिगार्डिंग द डिसीज. लेवल बढ़ जाएगा लिवर में, लिवर डिसीज में. विल सी द एंजाइम व्हिच आर एलिवेटेड इन केस ऑफ मसल डिसीज. लिवर डिसीज हो गया भाई, मसल डिसीज. दीज आर द एंजाइम व्हिच आर the level of these enzymes is increased in case of muscle disease. Creatinine kinase, first one is creatinine kinase. जहाँ पे भी creatinine kinase आएगा वहाँ पे muscle involved है. यहाँ पे भी है creatinine phosphokinase, ठीक है. तो myocardium भी क्या है, muscle ही है. Creatinine kinase is increased level of creatinine kinase there in case of muscular dystrophy. Remember muscular dystrophy, myocardial infarction, ये दो disease याद रखो. So increased level of this creatinine kinase is there in the body when you are suffering from muscular dystrophy or when you are suffering from myocardial infarction. Okay, then aldolase is the early marker for muscle degradation. Then aspartate transaminase also increased in case of muscle disease. Now bone disease में कौन increase होता है, alkaline phosphatase is increased in case of Puget disease and rickets. Both are the disease of bones. Rickets is caused by deficiency of vitamin D3 in the children. Osteomalacia is caused by deficiency of vitamin D3 in the adults. Puget disease also इसमें क्या है, bone malformation होता है, Puget disease. So it is a alkaline phosphatase. Now next is the pancreatic disorders. Pancreatic diseases में कौन से एंजाइम इंक्रीज हो गए, these two, amylase and lipase. They are increased. Amylase is increased in case of pancreatitis. Pancreatitis means infection of pancreas. So in that case amylase will be level will be increased. Lipase level will be increased in the blood in case of acute pancreatitis. Pancreatitis मतलब pancreas का infection अगर हो गया तो lipase amylase बढ़ जाएगा. Prostate cancer, प्रोस्टेट ग्लैंड का कैंसर अगर हो गया तो उसमें क्या है, ये एंजाइम का लेवल बढ़ जाता है. नेक्स्ट वन इज एसिड फॉस्फेटेज, अर्ली मार्कर फॉर एंजाइम फॉर प्रोस्टेट कैंसर. याद रखो अर्ली मार्कर फॉर प्रोस्टेट कैंसर दैट इज एसिड फॉस्फेटेज ACP. क्वेश्चन आ चुका है एक बार. प्रोस्टेट स्पेसिफिक एंटीजन दैट इज इट इज आल्सो इंक्रीज्ड इन केस ऑफ प्रोस्टेट कैंसर. याद रखो यहाँ पे हार्मोन are the enzymes which are increased in case of different diseases. So in case of myocardial infarction, these are the enzymes. So these are the enzymes. So their level in the blood will be increased. So remember cardiac troponin first is a early marker, lactate dehydrogenase is a late marker, and creatinine phosphokin is again early marker. HEPAT 22 में क्वेश्चन आ चुका है इसमें. Then hepatic disease में देखो ये चार एंजाइम है, alanine transaminase, aspartic transaminase, glutamyl, gamma glutamyl transpeptidase, and biochemistry. In case of muscle disease or disorder, ये तीन एंजाइम creatinine kinase, aldolase and aspartate transaminase इनका लेवल बढ़ जाएगा बॉडी में. बोन डिसीज में अल्कलाइन फॉस्फेटेज का लेवल बढ़ जाएगा. पैंक्रियाटिक डिसीज, पैंक्रियाटाइटिस, पैंक्रिया का इन्फेक्शन अगर हो गया तो ये दोनों का लेवल बढ़ जाएगा. अमाइलेज एंड लाइपेज. अगर आपको प्रोस्टेट कैंसर है तो उसका आपकी बॉडी में एसिड फॉस्फेटेज जो है वो बढ़ जाएगा. प्रोस्टेट कैंसर अर्ली मार्कर फॉर प्रोस्टेट कैंसर. नाउ यू कैन आई विल डेमोन्स्ट्रेट ओवर हियर. वी हैव सीन द mechanism of enzyme action. There are three theories. First one is nothing but the lock and key model or Fisher theory. So in which enzyme will have certain kind of configuration. Enzyme will have certain kind of configuration which is exactly opposite to substrate. So substrate and enzyme will have opposite configuration at the active site, and substrate will exactly fit on that configuration. That is known as the lock and key model. जैसे होता है ना, हर एक लॉक का एक ही की होता है, तो वो की उसी लॉक को फिट आता है और उसी लॉक वो लॉक ओपन कर सकते हैं उस की से। तो वैसे ही enzyme will have certain configuration at active site where substrate will fit. That is known as a lock and key model or Fisher theory. Okay. Now, second one is known as the induced fit theory. Induced fit theory. So, in this theory, this was proposed by this Coastland. Coastland's induced fit theory. So, Coastland has proposed that substrate will have specific configuration, but substrate will induce the configuration in the enzyme. जब substrate enzyme interaction होगा तो enzyme में configuration में change आएगा और बाद में substrate उसमें fit होगा. That is the induced fit theory. And third one is a substrate strain theory. So substrate will strain substrate will strain the enzyme and bring about the conformational changes in the enzyme, and that is a substrate strain theory. So this is just a theory of your. अब इतना याद रखो कि सिर्फ नाम याद रखो. फिशर थ्योरी मतलब लॉक एंड की मॉडल. ओके, फिशर थ्योरी मतलब क्या है? लॉक एंड की मॉडल. लॉक एंड की मॉडल क्या है? फिशर थ्योरी. इंड्यूस्ड फिट थ्योरी मतलब कोसलैंड्स मॉडल. एंड थर्ड वन इज अ सबस्ट्रेट स्ट्रेन थ्योरी. तो नाम याद रखो सिर्फ, बाकी कुछ नहीं. उसके बाद में एक और पॉइंट बचता है इसमें, एक्टिव साइट्स. फीचर्स ऑफ एक्टिव साइट. कि एक्टिव साइट में क्या क्या होता है? एक्टिव साइट में हमारे एसिड का सीक्वेंस होता है. ठीक है. एक्टिव साइट स्पेसिफिक होता है. एक्टिव साइट में इंटरेक्शन सब्सटेंस के साथ में कैसे होता है वो थोड़ा सा बाद में पढ़ लेना. दैट इज नॉट मच इम्पोर्टेंट. ठीक है, बट दैट इज ओनली पॉइंट रिमेनिंग. नाउ नेक्स्ट पार्ट इज सिलेक्टेड एग्जांपल्स ऑफ एंजाइम्स एंड देयर रिस्पेक्टिव सबस्ट्रेट एंड कॉम्पिटिटिव इनहिबिटर्स. एंजाइम देखो, xanthine ऑक्सीडेज नाम का एंजाइम होता है, उसका सबस्ट्रेट होता है xanthine एंड हाइपोxanthine. तो ये xanthine ऑक्सीडेज हाइपोxanthine xanthine को क्या करेगा? इसका कन्वर्जन करेगा यूरिक एसिड में. यूरिक एसिड में कन्वर्जन करेगा इसका xanthine का हाइपोxanthine का. तो xanthine oxidase is the enzyme. Hypoxanthine is a substrate, and by taking this substrate it will convert into uric acid. That uric acid will be increased in the body. Uric acid will form a crystals with sodium and calcium, and that crystals will deposit into your joints, synovial joints particularly, and there will be inflammation, edema and pain known as a arthritis, okay, or gout. So one inhibitor of this enzyme known as allopurinol. Yeah, allopurinol A, that is a competitive inhibitor of this xanthine oxidase, and it will prevent the formation of uric acid from this, and that's why it is used in treatment of gout. Now, another example, monoamino oxidase is the enzyme that metabolizes the biogenic amines, catecholamines. Okay, biogenic amines and catecholamines जैसे epinephrine, norepinephrine, adrenaline, noradrenaline, histamine वगैरह. That is metabolized by monoamino oxidase. Now what happens? The level of catecholamine is decreased in case of depression. तो कोई डिप्रेशन में होता है तो ये लेवल उसका कम हो जाता है. So in that case, monoamino oxidase inhibitors, they are used. They are used as a mood elevator or antidepressants. So for example, ephedrine, amphetamine, so they will inhibit the monoamine oxidase and prevent the metabolism of these catecholamines. So they are useful in the elevating the level of catecholamines in the brain in case of depression. Now another enzyme, dihydrofolate reductase enzyme. So its function is reduction of folic acid. dihydrofolic acid का reduction करना and tetrahydrofolic acid बनाना. So dihydrofolate reductase will convert dihydrofolic acid into tetrahydrofolic acid, which is very important for bacterial growth and cell growth. Okay. So there are certain inhibitors of this dihydrofolate reductase. Say aminopterin, amethotterin, and methotrexate, sulfonamides, etc. etc. They will inhibit the dihydrofolate reductase enzyme and prevent the formation of tetrahydrofolic acid, which is very important for cell metabolism. So this methotrexate, aminopterin, and amethotterin, they are used in the treatment of leukemia or blood cancer. Sulfur drugs, they are used as an antibacterial. Okay, they will act by same mechanism. And acetylcholine esterase is the enzyme. The enzyme responsible for the metabolism of acetylcholine. Okay, substrate कौन है उसका? acetylcholine है. It will metabolize the acetylcholine and convert. It will hydrolyze the acetylcholine and convert acetyl choline and acetic acid. Okay, choline and acetic acid. सो सक्सिल कोलिन इज द एंजाइम इनिबिटर, तो ये इसको इनिबिट करेगा किसको, सो एसिटिल कोलिन एस्टरेज एंजाइम इनिबिटर हो जाएगा, तो एसिटिल कोलिन का लेवल बॉडी में बढ़ जाएगा, which is desired in case of Alzheimer disease and also used in the surgery for muscle relaxation and muscle relaxation in anesthetized patient. Next enzyme is a dihydroterate synthase. So its substrate is a PABA, an aminobutyric acid, which is inhibited by sulfonamides. Then again vitamin K epoxide reductase. It is again very important. Vitamin K epoxide reductase नाम का enzyme है. Vitamin K epoxide reductase. Now this enzyme is coupled with the activation of your blood clotting factors. Blood clotting factors से जिसका activation होता है, those blood clotting factors which are vitamin K dependent blood clotting factors. Factor number 2, 7, 9, 10. है ना? So these are the blood clotting factors known as vitamin K dependent blood clotting factors. उसके activation के लिए ये enzyme लगता है. Vitamin K epoxide reductase. उसका substrate क्या है? Vitamin K. Vitamin K is the substrate. और उसका inhibitor कौन है? Dicomerol. Dicomerol inhibit the vitamin K epoxide and prevent the activation of blood clotting factor, and that's why it acts as an anticoagulant. ये आपने देखा है already when we was discussing about the anticoagulant. Now HMG CoA reductase एक एंजाइम होता है हाइड्रोक्सीमिथाइल ग्लूटारिल CoA. That enzyme hydroxymethyl glutaryl CoA reductase. It is a very important enzyme in the biosynthesis of cholesterol. ठीक है, जो कोलेस्ट्रॉल होता है बॉडी में बायोसिंथेसिस, उसके लिए एंजाइम लगता है. उसका सबस्ट्रेट क्या है? हाइड्रोक्सी मिथाइल ग्लूटारिल CoA. तो दिस इज इनहिबिटेड बाय लोवास्टैटिन, परवास्टैटिन एंड स्टैटिन्स. जो स्टैटिन्स होते हैं दे आर नोन एज एंटी हाइपरलिपिडेमिक ड्रग्स और हाइपोलिपिडेमिक ड्रग्स. दे इनहिबिट द एंजाइम HMG CoA reductase and inhibit the de novo synthesis of cholesterol or biosynthesis of cholesterol. So that's why these are the selected enzymes, their respective substrate competitive inhibitors. Now we'll see the next. So enzymes, coenzymes of big complex vitamins. अपन ने coenzyme देखा है. coenzyme मतलब क्या? Non protein part of your hollow enzyme. A coenzyme is a protein part of your enzyme. Okay? Coenzyme. So यहाँ पे देखो लिस्ट है coenzymes. विटामिन बायोसिंथेसाइज फॉर्म विच विटामिन क्या ट्रांसफर करते हैं और इसके ऊपर कौन सा एंजाइम डिपेंडेंट है. सो कोएंजाइम derived from vitamin, atoms group transferred by that coenzyme and the dependent enzyme. Okay? So coenzyme का नाम है थायमिन पायरोफॉस्फेट, TPP, थायमिन पायरोफॉस्फेट. So that is biosynthesized from, that is derived from thymine and involved in the transfer of aldehyde or ketone group. Okay? And TPP dependent enzyme was by this transketolase. So this is the enzyme which is dependent on coenzyme known as a TPP, thymine pyrophosphate. क्या करेगा ये? aldehyde group को ट्रांसफर करेगा. थायमिन पायरोफॉस्फेट में थायमिन विटामिन होता है. देन फ्लेविन न्यूक्लियोटाइड या फ्लेविन न्यूक्लियोटाइड, इसमें विटामिन कौन सा है? राइबोफ्लेविन है. और ये कोएंजाइम क्या काम करता है? ट्रांसफर ऑफ हाइड्रोजन and electron, transfer of hydrogen and electron. So these enzymes, L amino acid oxidase, D amino acid oxidase, L. Okay, so L amino acid oxidase is dependent on this coenzyme flavin mononucleotide, flavin adenine dinucleotide. So it is also obtained from riboflavin, catalyzes the reaction of hydrogen and electron transfer. Okay, again D amino acid oxidase is the enzyme dependent. तो एक लिस्ट है बायोकेमिस्ट्री बुक जो है उसमें है ये. मैं यहाँ पे एक्सप्लेन एक दो दिन ऊपर के दो दिन किया है, बाकी आप पढ़ लेना है. This was the today's session. तो अगर आपको ज्वाइन करना है, यू कैन ज्वाइन आवर ऑनलाइन कोर्सेज by paying, and same lecture whatever I am taking live, that will be also